This is the BBC.
This podcast is supported by advertising outside the UK.
BBC Sounds. Music, radio, podcasts.
I have to say, this is looking rather nice. A nice, good, deep brown topsoil profile here.
Looks like cocoa, doesn't it? The colour
It
of cocoa.
does.
It's beautiful. You're pleased with that then, Chris?
Really pleased. It's got a really nice crumb structure to the soil. It's not coming out like anything like muddy at all. And so we're down now a good spade depth, and the soil hasn't even begun to change colour yet.
This is Chris Jones, who runs Woodland Valley Farm, nestled in Laddock Valley in Cornwall. Chris is a friend to the microbes that live in his soil, and with good reason, because it's no exaggeration to say that microbes are the foundation of life on Earth.
It's beautiful. You're right, actually. If you run that in your hands, and it's like the perfect crumble texture if you're making a lovely apple crumble.
And just have a little smell of that.
Okay, let's have this. Oh, why is that? Oh, yeah, so that is that earthy smell.
Yeah. The thing about the smell is that if your soil is not healthy, it tends not to smell healthy. Whereas this is just a beautiful, deep, but kind of light soil aroma. I can't describe it any better than that.
Well, it definitely makes you take an extra deep inhalation, almost like you're trying to savour a fine wine, but this is a olfactory sense, a smell. Yes, lovely.
It's often said that just a teaspoon of soil contains more life than all the humans on Earth. Microscopic life, that is, bacteria, fungi, protozoa, nematode worms, and microarthropods like springtails and mites. But there are signs that this extraordinary invisible world is in trouble.
There is increasing evidence that the Earth's microbiome is under threat. We know most plant species have plant species-specific microbiology. When plant species go extinct, so does their associated microbiology. When we look at our global change experiments, so places on the Earth where we experimentally change how warm it is, how wet it is, the frequency of drought, we're seeing a remarkable decline in microbial biodiversity here as well. Taken together, these early signatures strongly suggest an impending major decline in microbial biodiversity on this planet.
I'm Gillian Burke, writer, biologist, and presenter. And believe me when I say I love soil. I grew up in Kenya, on the outskirts of Nairobi, where the soil is this beautiful ochre red. And as a child, I loved playing with soil. I'd even eat it sometimes. Over the years, I've discovered what actually lives in soil, which, far from put me off, has only made me even more fascinated with the stuff.
So, when did you, as a farmer, become aware of this invisible, trillion-strong community working away in the soil?
I suppose in a very serious way about 15 years ago. We had a carbon audit done, which sounds a million miles away from bacteria, but we had a carbon audit done, and the overall farm was emitting a net emission of carbon, and I thought, gosh, that seems a bit odd when we're such a tiny farm, and it's just a little eco-friendly organic farm, how could this be? And then I learnt about cultivation and exposing soil to the open air, and this loss, this oxidation of organic matter resulting in the carbon emission. And then we learnt that if you didn't do the ploughing, then the soil would begin to accumulate carbon. And of course, that carbon accumulation is the microbiology. That's what it is. So if you've got a really, really active soil that you're not cultivating or over-cultivating, there's every chance that it will become a real carbon sink. So that's what got me into it. This knowledge that we gained from the carbon audit has helped to completely change the way we do things here. Because we have this a beautiful chicory plant just here, you can see it's got these big roots here. Now these roots will extend down to about 1 metre, and these deep tap roots, they are getting into areas of soil which may not have any other roots in them, because your grasses and so on tend to be not so deeply rooting. And what that means is you're getting air and food getting down into the soil, because what feeds all this soil microbiology is photosynthesis. The plant is growing and it's photosynthesizing during the spring and summer and autumn. In fact, in our context in Cornwall, probably all year round to an extent. And a good deal of the plant food that's being created in that photosynthesis is ending up down in the roots and then feeding lots of soil biology adjacent to those roots through what they call root exudates. And that is keeping a very, very healthy and abundant soil biology going.
So those are the exudates, those are sugars, bit of carbohydrates and protein. So that's the stuff that the plants are making and feeding these microbes in the soil.
Yeah, exactly. Prosper.
As if we never were here. Lovely.
Colin Averill is a senior scientist at ETH Zurich and the founder and CEO of Funga. I was fascinated to read about his work introducing healthy fungal microbiomes to new growth forests, and I wanted to find out more.
I really liken the work we do to that was, the concept started by the Human Microbiome Project. So each of our bodies is a microbial ecosystem. There's an incredibly biodiverse community of bacteria in your gut that we now know has profound effects on your health. And just like the microbiome can affect your body's health, the microbiome of a forest and the microbes that live in the soil, their identity and their biodiversity, can affect forest health. It can affect the ability of trees to grow, how fast they grow, how much carbon they store in tree stems and in soils, and it can affect their ability to resist drought. There's a huge variety of things that this microbiome can affect in a forest. And the questions we're beginning to ask and answer today are, given the way we've managed ecosystems historically, and especially soils, is the microbiome degraded? Have we lost significant biodiversity? And when we go to plant a forest to do restoration or reforestation, is it missing its microbiome? And what would happen if instead of just planting the tree, we also planted, quote unquote, the microbiome?
Okay, because when thinking about a piece of agricultural land, it's easy to make that mental leap that the microbiome has been disrupted, harmed, degraded as a result of inputs, whether it's fertilizers or pesticides or just soil erosion. How would that take place in an environment that hasn't been farmed, an environment where there may be already some forestation? Like, just describe that process of how it got degraded in the first place.
There's two big culprits here. One is a lot of our forests, especially in Europe, are intensively managed, and they're managed for harvest. And when those clear cuts happen, there can be a huge disruption of below ground biodiversity. And that's because many of these microbes that live in the soil are like biological partners, so they're in symbiosis with the trees. They rely on a supply of carbon from photosynthesis from the tree. When you kill the trees, they can just wither and die, and that's really well documented. And so when reforestation happens later, many of those microbial community members are now gone. And so that's one way we lose them. Another way that's been really well documented is air pollution. So historically, through burning fossil fuels, we don't just emit carbon dioxide, we also emit nitrous oxide, and that rains down on ecosystems as essentially nitrogen fertilizer, which can actually massively shift which fungi partner with the roots of trees. So those are two key ways. And I guess the last thing I'd mention is that when we do reforestation, most of that happens on ex-agricultural landscapes. So there was a forest, it got cut down for agriculture, and now we're trying to put it back. But that soil, because it's been agriculture for decades often, it's lost a lot of those forest-specific microbial community members.
Okay, so it's interesting you use that forest-specific. I'm assuming that no two microbial communities are the same as you move across a landscape. Is that right?
That seems to be true.
Okay, so can you just explain a bit more about how that view might have changed, like from the early days of soil microbiology? What have we learned in terms of the variability, as you move across a landscape and from, even one continent to the next?
Going back like 100 years in microbiology, there's something called the Bos-Becking hypothesis that came from a microbiologist called Bos-Becking. And he made this statement, everything is everywhere, but the environment selects. And what he was referring to is he would put out petri dishes and he would collect microbes in different parts of the landscape of the world. And he'd always find the same ones, but which ones persisted depended on the environmental conditions of what's the pH of the media and different things like that. He concluded okay, these microbes are so small, it's so easy for them to get around. They're really just everywhere. And so there's no sort of microbial biogeography to speak of, right? There's no sort of terroir of the microbiome. That started to change a lot when we started using DNA technology, DNA sequencing technology to study the microbiome. So basically, the Human Genome Project created all this new DNA sequencing technology that environmental microbiologists co-opted to study microbes in the environment. And so we started being able to create DNA-based profiles that tell us which bacteria or which fungi live in this soil or that soil or that soil. And one of the first things environmental microbiologists looked for was sort of these distance decay relationships. So if you look at plant and animal communities, the further away two locations are, just the more dissimilar two plant communities are. And what people first observed was that there was remarkable distance decay in these microbial communities. And that's also really supported by really old studies from the early 1900s. Some of the best stuff was done with rhizobia. So when people grow soybeans, they depend on these symbiotic nitrogen-fixing bacteria that live on their roots. And it was well known that you could plant soybeans and they'd grow very poorly. But if you brought soil from an existing soybean field that was performing well, you could improve performance. And it was because it was reintroducing those key symbiotic bacteria to that field.
Okay. So you've described really beautifully how even in the early 1900s, people understood about bringing soil from an established plantation to establish a new soy plantation. In the context of reforestation, what does that look like today?
Here, it's really helpful, again, to draw an analogy to the Human Microbiome Project. We know that the human body is a microbial ecosystem. So each of us has an incredibly biodiverse community of bacteria that lives in our gut that has a profound effect on our health. And we've also learned that if you are sick because you've lost key components of bacterial biodiversity from your body, you can treat some of those conditions by doing microbiome restoration. Could we go to a forest that we think harbors great below ground microbiology, and could we take the soil? Because that's where digestion happens in the forest. That's the microbial rich material. And could we transplant that? So instead of just planting a tree, we plant a tree and we quote unquote plant its microbiome. And so we started testing this idea in Wales in the United Kingdom in collaboration with a charity there called the Carbon Community, run by Charles Nichols.
So we're standing in here in a piece of beautiful mid-Wales on a lovely sunny day. And we're surrounded by about 170 acres, which has been planted with around about 100,000 trees. And within that, there is a new scientific field trial, which is really looking at how to help trees grow faster and sequester more carbon. And my name is Charles Nichols from the Carbon Community. So really what we're doing is we're looking at what happens below ground as much as what happens above ground. And below ground is really important because when you look at a tree, there's a big oak up there on the ridge line, the bit that you can see is only 22% of the carbon that's stored. The bulk of it is stored in the roots and in the soil. And fundamentally, there is a relationship between what happens below ground, in particular with mycorrhizal fungi, that then help the trees to then absorb the nutrients from the soil. In return, they get sugars back. It's one of those relationships that's fundamental to the way that the world works. 80% of plants rely on this. So one of the techniques we're using is we're applying crushed basalt. It comes from a mine about 20 miles up the road, so not very far away. And basalt is a very widely occurring volcanic rock. It's mainly used in the manufacture of asphalt for roads. But when you mine it for those little chips, it produces lots and lots of dust. In fact, at the mine up the road, 200,000 tons of this dust sitting around that they're not quite sure what to do with. But in fact, what we do is using a lime spreader, we spread it on the fields before planting, and then when rainwater falls on it, it absorbs carbon dioxide through a process of chemical weathering and then releases nutrients for the trees. A little bit like a vitamin shot. If you come over here, I'll just show you. So next to every tree, we have a stake, and on the stake is a barcode. And the idea is basically that this gives this individual tree its specific identity. And we use an app on a phone, and we get volunteers along. They scan the barcode, and then, probably working in pairs, they will measure the diameter of the tree at the base, and then they'll measure the height to the tip. And if the tree is big enough, it's over 1.3 metres, then they'll also measure it at 1.3 metres. And that height at 1.3 metres is a measure that scientists can use to then estimate the overall volume and height of the tree. In this scientific study, we're actually testing two different types of forests, so monoculture conifers, which is sort of similar to the way that commercial forestry works, and broadleaf planting, which is more typical for restoration planting, and then 4 different kinds of techniques. And to do this in a really robust way, because we're testing these things in combinations to find out what combinations work best, we've actually then built a study where we have 8 test cells and a control group, and that's replicated 8 times, giving us a total of 72 test cells. This makes it one of the largest, most complex, maybe even crazy projects out there, because what we need to do is, within each of those cells, we need to every year measure the health and growth of individual trees, and it is a massive piece of work. In 8 days we measure 6,400 trees. That's about 200 person days worth of work. I mean, the trees have been in the ground now, they're in their 3rd growing season. At the end of the first year, we noticed or observed through all of these readings that we've been taking, some of the trees that have been with some of the treatments we've been doing are growing maybe 30% faster. And this isn't about the trees that we plant here. The big idea really is, well, could we help all trees everywhere to grow faster and sequester more carbon? And that's a huge deal. If you plant on average 15 billion trees a year, and you could get them all to grow 30% faster, then you could absorb 450 million more tons every year just by doing the same thing with a little bit of extra effort. So the potential is enormous. We've now really got to figure out how to deploy this at scale, and that's really what we're doing here. This one hasn't flushed yet, so I'm just checking to see whether he's fine. He's absolutely fine. He's coming. It's just a little bit of a late starter. So these are all my babies.
It's not just how soil microbiomes benefit us. What really fascinates me is their world of almost infinite connections. A whole invisible world of over 1 trillion species, so vast we might as well be talking about a universe.
Bacteria first appeared on this planet 4 billion years ago. Fungi have been around for, on average in general, about 3.5 billion years. And Mother Nature doesn't keep feeding something that has no purpose. So if we were really in a situation where bacteria and fungi, protozoa, and nematodes were pointless, they weren't helping the plants at all, Mother Nature would have thrown out that dead end, wouldn't she? And yet here they are, 4 billion years later. Just because us human beings are too stupid to see something that's smacking us in the face doesn't mean it's not real.
One person who understands this world better than most is Dr. Elaine Ingham.
The soil food web is going to include bacteria and fungi, protozoa and nematodes at the minimum, because the interactions between these groups of organisms are what will provide nutrients to the plant in a plant available form. And I think that something that most people don't recognize is that when you're talking about sand, silt, clay, rocks, pebbles, parent material, all of that sort of thing, that's not available to your plant. You have to have the predators come along and eat the bacteria and fungi and have those critters, the predators, say, I've got too much nitrogen, I've got too much calcium, I've got too much, whatever it is, and it's going to kill me if I don't get rid of it. So they've got to poop it out. And of course, where is all of this happening? It's right in the root zone as these nutrients, now in a soluble form, which is the form that plants can take up.
So can I ask a question? What is soil without all of that life in it?
It's dirt. And so that's the difference between dirt and soil. Is dirt is just the mineral component, the sand, the silt, the clay, rocks, pebbles, etc. But you're not going to get any benefits out of that inert material just sitting there, which means now you've got to go and buy inorganic fertilizers, you've got to buy pesticides. We have to do what nature has always done and put together the proper biology. When we have people that we work with going out and teaching them how to use this system, how to make really good compost so you have all of the organisms, how to apply that so it's very easy on the farmer to be applying. It doesn't cost much. We increase yields by somewhere around 100 to 300%. Plus, it's got all the nutrients. Will help you as a human being be healthy yourself.
So healthy soil equals healthy bodies.
Absolutely. You cannot farm properly if you have less than 3% organic matter in the soil. You don't have enough food to keep the bacteria and fungi alive that the plant must have in order to get the nutrients it requires and then balances it requires. So, yeah, we have done a pretty good job of, turning things around, getting more people understanding they got to go down this pathway instead of the pathway to hell.
Colin Averill.
Why do we care if a snake you've never met that only lives on a mountaintop in Guyana goes extinct? I think we'd all agree that sucks, even though it doesn't do anything for us. And I think that's also true of the bacteria, the fungi, the mold, the microorganisms.
We've heard about how the microbiomes in soil can be nurtured and how transplanting the microbes from healthy soil to unhealthy soil can help trees. But what about the microbes themselves? Well, unsurprisingly for organisms that have been around for millions of years, they're very, very resourceful. I describe it, we're not far from the sea, we're never far from the sea in Cornwall, but we're at Paul Dice Mine in an area that I guess for me, I think of as like Cornwall's mining spine, an area that has been worked very hard and extracted for centuries. So I'm with Dr. Elsa Hesse from the University of Exeter, evolutionary ecologist. And Elsa, you've been studying this site. Why?
So mining is a problem worldwide. So we've got a huge legacy of mining waste covering large areas of the world, also quite often in populated areas. And these mining materials are often toxic to humans, but also to wildlife and plants. So my research tries to find ways to clean up mining waste in a sustainable way using microbes.
Using microbes. Now that's interesting because this has obviously been here a long time. These are essentially the ruins of Cornwall's mining industry. So just to kind of go to the first principles, because I'm going to be a bit sort of simple here. I mean, presumably microbes aren't doing this for our benefit. Why would microbes be able to handle novel toxic waste that they wouldn't have evolved alongside with?
So these toxic waste are also not only toxic to us, but also to the bacteria. And some of these mechanisms do not only confer resistance to metals, but also to other toxic compounds. So when you end up in a toxic environment, you will select for a subset of microbes that have evolved to deal with these toxic substances.
So would these bacteria, prior to the mining and obviously all the toxic metals leaching now into the environment, would they have existed before that?
So I think the metals would have selected for bacteria that were better able to cope with toxicity per se, and then they've actually adapted to the toxic metals to make this whole process more efficient. So where we're standing here, it has a very special subset of bacteria that are uniquely able to actually deal with toxic metals. And as such, the composition of these communities differs a lot from any other soil sample you can find in the UK.
From a standing start, how long would it take a microbial community to develop this ability to handle this toxic waste?
So, depending on the environment they're living in, they could evolve metal resistance within a matter of days in the lab. How that works out in the natural environment is unclear, because they tend to be a bit resource poorer, meaning that population sizes are smaller.
That is absolutely amazing. I wasn't expecting you to say a matter of days. I know that's in the lab, but that indicates how quickly they turn over in terms of generations, how quickly they can respond to these changes in their environment. So in a sense, like when I hear this, it's encouraging. You're like, well, there is this mining legacy. We need to be able to do something about it. And actually, we almost don't, because the microbes have done it themselves. But looking ahead and going forward, do you think there's a danger that industry then sort of falls back on this ability for microbes to deal and clean up their mess? And what does that mean for the future of like mining?
So just to emphasize, so microbes, they deal with the toxic metals, but usually they deal with it in such a way that it's still present in the soil. So we need ways in which we can use their capacity to actually extract the metals from the soils. And we're still quite far off that. So there's no clear-cut solution at the moment, and there still needs to be quite a lot of research on this topic. So I think it's very promising that we potentially could use microbes, but there's still a long journey ahead of us.
Our understanding of soil microbiomes may still be a work in progress, but we do know enough to know that the impact of industrial food systems